Hope everyone can hear me okay. Can Nadia or Noemi give me a thumbs up? We're good? Okay, everybody can hear me. Great. We're going to give it a couple of minutes to let a couple more people trickle in here, and then we'll get rolling. So welcome. Lots of people filing in right now, which is great. All right, let's go ahead and get rolling. So I want to welcome everyone and just say thank you very much for attending this webinar. We've got some really exciting topics to cover today. I want to give a quick introduction to myself and our guest speakers. So my name is Jonathan Fisher. I'm the Senior Vice President of Commercial for Akoya Biosciences. We're happy to host this webinar with LabRoots as a partner. And I'll be moderating the webinar today. I'll be giving a quick introduction to our guest speakers before we sort of kick things off. And the agenda will run as follows. So I'm going to give a quick couple of slides on our latest platform from Parhelia, which will be folded into the talks given by Nadya and Noemi. And then once I've done that, my very, very quick piece, I will hand it off to Nadya, and then she will hand it off to Noemi. Now, a couple of housekeeping items here. We want to make sure that everyone sees that there is a question and answer section, so you can submit your questions throughout the webinar. That's going to be in the bottom left-hand corner. You'll submit your questions. We'll get to those at the end of the presentations. If you have a particular question for Nadya or Noemi, if you'll please key that in, that is for them. If it's with their slides, they can roll back through their slide presentations and we'll have the correct person answer it. Something else that's really important that we want to make sure that we cover as well is we do have a poll question that's a survey off to the right-hand side of the screen. If everyone could please submit that, you can select multiple entries for that. I would imagine that a lot of people are going to select multiples, but that just helps us and helps us drive future conversations and future innovations that we're going to bring to you from Parhelia. So with that said, what I'm going to do is I'm going to go ahead and introduce our guest speakers, and then I'll go ahead and take and present a couple of slides. So our first speaker behind myself is Nadya Nikulina, and currently she is a researcher and PhD student at Max Planck Institute, JLU University in Gießen, Germany. She's one of the world's foremost experts in the Phenocycler Fusion technology, formerly known as CODEX. So she was and has the distinction of being the first employee of Akoya. So she's probably run more and seen more Phenocycler Fusion data than anyone else potentially in the world. So we're very happy to have her on the call and having her present. She will be presenting some Phenocycler Fusion data, which is fantastic. So if you are running that assay, this might be your time to chime in and talk to one of the world's leaders in that. And she spent 8 years at Akoya helping develop the applications there. So our next speaker is Noemi Kedei, MD. She is a staff scientist and head of the Spatial Imaging Technology Resource at the National Cancer Institute, NCI. She is quite a resource. If you've ever visited her lab or ever seen her present, she is very knowledgeable in a variety of spatial technologies, including but not limited to Phenocycler Fusion, formerly known as CODEX, GeoMx DSP, CosMx SMI, in addition to a host of others that I know of. So we're happy to have her on the phone as well and to present some of her data and findings also. So with that said, I'm going to launch right in. So giving you a little bit of a reasoning behind why Parhelia was started and sort of the latest generation offering that we brought to market in a robot platform. Our company was founded by Nikolai Samusik and Yuri Goltsev, and they were also founders of Akoya. You know, great chemistry, great technology, great imaging devices. One of the things that they saw as a gap in spatial was the need for an integrated solution that covered sample prep. Sample prep for us covers a number of areas. As you can see here, we sort of categorize it into 4 buckets: DWAX and epitope retrieval. You have morphology or H&E staining, and then you also have staining and hybridization of proteomics and transcriptomics. So we needed something that was more of a universal platform that was creating sort of an end-to-end sample prep automation. Now, what does that do? It allows you to automate and step away and not be married to a Coughlin jar, a hood, and a pipette. But it also downstream helps sort of minimize variability in data and streamline analysis for the folks that are doing the analysis on the back end. So it really frees up your time and improves your data quality that you receive from spatial. And it gives you the flexibility of going higher or lower throughput depending on what your need is. You're not hamstrung by having only 2 hands. So we launched earlier this year at AACR this Spatial Station, which is a Parhelia platform that we say is the next generation platform for spatial biology. With that said, it was purpose-built. There are other platforms in the market that claim that they can do sample prep for spatial biology. What I find is that people use them more so as one-trick ponies for one thing or another, and they don't like what it does. For instance, with staining, it might utilize way more reagent than you'd like, or it has limitations in heating and cooling. There's a substantial amount of dead volume, which leads to increased cost per run, etc. So what we aim to do with designing this robot was, 1, make it purpose-built for spatial biology. 2, make it a benchtop instrument that doesn't need a room to house, and make it very, very user-friendly. We knew that spatial biology was sort of a budding industry, and we expected to see new chemistries and new protocols hit the market, and we wanted to be flexible enough to encounter those and then master those very, very quickly with the platform, even just in the customer's hands. So we aim to be completely assay agnostic. So we can take any sort of chemistries or assays and actually, if it's moving reagents around, heating, cooling, and doing all those sorts of things that are really critical, and do them very, very high precision. Again, we wanted to enable sort of an end-to-end. So you're not just doing a baking and then dewaxing where you're running to the hood with silenes, and then you're using a pressure cooker for epitope retrievals, and then you're staining by hand in Coughlin jars. We wanted you to be able to set up a platform and then walk away from it. So it does the baking, dewaxing, epitope retrieval, staining, hybridizations completely in a line. You can go to lunch, you can analyze data, you can go home for the evening. That was kind of the idea. The platform also has great sample heating and cooling. Most platforms stop at about room temp. We go down to 0. So we can do this 4-degree hybridizations, which you'll probably hear about Nadya's talk, that are critical for Phenocycler Fusion. An ability to go up to 100 was also critical, especially if you're going to do epitope retrievals. So something we wanted to incorporate. And we wanted to, again, make it so that as we proceed through the spatial biology market, we wanted to allow clients to go in and integrate assays as well. We're seeing a lot of folks that are starting to move into proteomics and wanting transcriptomic data from the same slides. That is something that's very achievable with these robots. In addition to that, one of the very common things that we're seeing is most groups require an H&E and a morphology stain, as well as to go alongside their spatial biology stains. So that's something that's very achievable, and you'll actually see that on the talk today. So with that said, that's sort of my piece. Again, if you have not answered on the poll or the survey questions on the right-hand side of the screen, please do. And you have questions on the left side of the screen also. If you'd like to submit those questions throughout the talk, we'll come back to those at the end. Thank you very much. And I'm going to go ahead and hand over to Nadya and let her take it away.
Thank you, Jonathan, for the introduction. Good morning, good afternoon, and good evening, everyone. I will be presenting automation of Phenocycler assay using Parhelia Spatial Station today. And first, let me start with the overview of Phenocycler Fusion workflow. The Phenocycler Fusion platform, it's a platform that was developed at Akoya Biosciences. It's a benchtop spatial biology solution enabling high-plex imaging on a whole tissue at single-cell resolution. Today, we will be focusing on a sample preparation for Phenocycler Fusion assay. So, basic things that you need to know how the Phenocycler Fusion assay works. You have antibody that conjugated to unique oligos, we call them barcodes, and you have corresponding reporters for each of these oligos. These corresponding reporters are tagged with a different type of fluorophores. So what is important in Phenocycler Fusion assay that you stain your sample once with entire antibody cocktail, you fix your antibody on a tissue, and after that, your sample goes in Phenocycler Fusion platform. The Phenocycler workflow consists of multiple cycles of revealing, imaging, and removing fluorescent reporters. During each cycle, 3 fluorescent reporters are tagged to their corresponding barcoded antibodies and imaged. After that, the 3 reporters will be removed and cycle will repeat again. This is a quick overview of Phenocycler Fusion workflow. Recently, I think in July, the Phenocycler Fusion protocol, actually, it's a really nice paper, was published in the STAR protocol by Cell Press. You can read this paper and you will see that it's described very in detail each step of Phenocycler Fusion workflow. And it's actually very nice process diagram that you can see on the right side. That you can see it's a multiple steps assay. And today we will focus on step number 1, number 2, number 3, and number 4. So actually all these 4 steps that can be fully automated on Parhelia Spatial Station platform. And I want to talk about how to adapt this manual process and adapt this process and basically utilize the automation to make our life easier. And when we take a look at the manual protocol, I highlighted over here the timing for each step. And you can see that usually it will take around, like, probably based on this paper, it will take more than like 20 hours of manual steps. I want to indicate that on step 2, when they were doing antibody staining, it was overnight staining, but still, all on other steps, basically, you need to be present at your lab bench to perform this staining. You know that when you're working in the lab, when you're doing protocol manually, it's always can lead to errors. We're all humans. We have different type of days in our life. And also it depends on the training, how good, how qualified the scientists or technicians are in the lab. Because even though this protocol is pretty straightforward, I would say, but because it includes a lot of steps, it's very easy to make mistake. And what this type of mistake usually lead to us, it's repeating the experiments. And you know that running experiments, it's not cheap. It's always, especially high-plex experiments. The high-plex experiments are expensive. And obviously, with the manual protocol, on each step, we are using much more reagents comparing, for example, to the Parhelia Spatial Station. That's why, personally, for me, I would say it was game-changing when we could automate all the steps in Parhelia Spatial Station. Basically, how much time it takes for you to start your experiment. Usually for step between 1 and 3, you need only 20 to 30 minutes to set up your buffers, basically aliquot just your reagents. And for step number 4, for reporter plate preparation, again, just to set up reagent might take only 10 minutes. You can see that we're using our time very efficiently. That it's a very good thing, because obviously time is money. Plus, we're not present. It's all robotics based. So it means that we are reducing possibilities of any human errors. Plus, I want to emphasize that each step of Parhelia Spatial Station, when we are running the protocol, it only takes around 150 microliters per step. So it's a significant reagent savings as well. And to just summarize this, I would say that when we are doing manual protocol, we're probably not always utilizing our time correctly. A lot of times the manual protocols can lead to inconsistent results, and obviously it's a high risk to repeat your experiments. On the other hand, when you're running automated protocol, you will be more focusing on more high value activities. For example, for me, it was always the QC of my data, and it's more consistent and reproducible results. And also we are reducing obviously risk of repeating experiments, and we are saving money. So in this slide, I want to highlight that today we will discuss the part of the staining protocol for Phenocycler assay. And you can see over here that Phenocycler assay includes a lot of steps. All the steps we will successfully transferred to Parhelia Spatial Station. I want to indicate 2 particular steps over here. It's antibody mix preparation and fixation step after antibody incubation is done. After antibody incubation is done, we have multiple fixation steps with PFA, methanol, and with fixative. These type of steps are extremely crucial for Phenocycler assay. And the reason is because we want to make sure that our antibody is attached properly to the tissue. Since our samples will go to Phenocycler Fusion platform, and we will have multiple washes. We want to make sure that antibody is attached. We will not lose any signal during our multi-cycle experiment. So after you perform your Phenocycler Fusion experiment, you also have an option to remove the flow cell. Removing the flow cell, it's a manual process, unfortunately. But after that, you can perform H&E staining. And H&E staining, you can perform on Parhelia Spatial Station as well. And I will share post-PCF H&E staining with you in the next few slides. So, but before that, let's talk about a little bit about this fixation step. I already mentioned that fixation steps after antibody incubation, it's extremely crucial for us. And when we were transferring basically manual protocol to Parhelia Spatial Station, we wanted to make sure that all the steps that we're implementing at Parhelia Spatial Station, all fixation steps, are correctly implemented. And we don't lose any antibody during multi-cycle experiment. What was done? Here, we took one antibody, but the panel was like a 15-plex panel, and this is one of the antibody in 15-plex panel. So this antibody is CD8 conjugated to BX026. with corresponding reporter Rx26 on out of F50. Exposure time during all experiment was exactly the same, again, because it's exact same tissue. Obviously, ratio of the antibody is the same as well. So what we did, first we perform our 7-cycle experiment on Phenocycler Fusion. Again, the staining was done on Parhelia spatial station. After the experiment was completed, we took our sample and we store our sample at 4 degrees in a storage buffer for 48 hours. After that, we took our sample and we put it back on Phenocycler Fusion platform. And again, we ran Phenocycler Fusion experiment. It was exact, obviously same panel, and we used exact same reporters from the same vial. So what you can see here on the images from experiment 1, we put our antibody, our reporter on a cycle 2, even though here it's indicated first cycle, but it's a first cycle with when we are actually detecting all our targets, because if you know that cycle 1 in Phenocycler Fusion, it's always a blank cycle for background subtraction. So, okay, on the first cycle, we were able to detect CD8 antibody. And over here, you can see the image and staining quality of CD8 antibodies. After that, on the experiment 2, again on the first cycle, we added reporters for CD8 antibodies, and again, we were able to detect CD8 antibodies. Also, on experiment 2, we added reporters on the last cycle for CD8 antibody. So, in ideal scenario, and actually this is ideal situation, we should not be seeing any loss of the a signal. And you will be able to detect each cell. And over here, I choose these 3 cells that they are separate from each other. So, and I did little quantitative analysis that you can see on the next slide. What you can see over here, if we will take a cell 1 on the first cycle in experiment 1, again, cell 1 on experiment 2 in the first cycle, and cell 1 on experiment 2 in the last cycle. You can see the numbers that they are pretty much the same. And the same thing exists for cell number 2 and cell number 3. So it means that our fixation steps are correctly performed and implemented on Parhelia spatial station. We don't see any loss of the signal. We were able to detect exact same cell and pretty much exact same level of intensity. Next thing that we wanted to do, and we did successfully, we wanted to compare our manual protocol and automated protocol on Parhelia spatial station. What was done? So we had close to serial section human FFPE tonsil. Our tissue, we had 2 slides, one for spatial station and another one for manual protocol. Both of these slides went through the same dewaxing, tissue rehydration, and antigen retrieval steps. It was no variation over there. After that, one slide went through manual staining protocol, and another slide we put on Parhelia spatial station. However, we used exact same reagent, and we prepared the antibody cocktail that we used for manual and spatial station sample. Also, both of these samples were imaged within 24 hours. So it's a minimum variation or impact of the storage condition. And I was trying to select region of interest that are very similar morphologically. Here we have small images of, I believe, 12 different antibodies. And in the next few slides, I will go through a few of them in a large format. So for this experiment, Actually, all the settings on manual and spatial station, they're exactly the same, like a brightness contrast. We use exactly the same exposure time. Obviously, it's the same antibody cocktail that we used for the both manual and spatial station. And you can see, and you can visually compare manual staining with spatial station staining. This antibody is pancytokeratin. Again, I was trying to choose a similar region. And we were also trying to choose different antibodies on the different channels and like immune cells targets, structural components target that you will see in the next few slides. Next, the CD3 antibody. Again, the settings are all the same. Next one, CD31, beta-actin, CD8, CD20 for B cells, CD45. Over here, we observed that staining on Barhelia spatial station was actually quite saturated. It's in a way good thing. You can use less antibodies. CD68, CD206. CD206, I want to emphasize that this antibody was internally conjugated. They're not part of Akoya commercial antibodies. So we wanted to include different type of antibodies, commercial antibodies and internally conjugated antibodies, since Akoya platform gives us an opportunity to conjugate antibody of our interest. Ki67, and on the next slide, you can see the combination of different targets, structural targets for epithelial cells, for example, pancytokeratin, for B cells, for T cells, CD3 and CD8, endothelial cells, and CD206 for macrophages. And you can see also in large regions, and you can visually compare the manual samples to Barhelia spatial station sample. So one last thing that we wanted to check also, and in a way challenges the system. Very often, a lot of us work with TMA, or we put multiple tissues on a one slide. And it's very important that your staining will be like evenly distributed, your antibody and buffers will be evenly distributed, and you will not see the like a weird staining pattern between the tissues, between different tissues on your slide. So what we did basically, we took a slide with two FFP tonsil tissues on the same slide. This top tissue, it's more close to slide label, and the bottom tissue, obviously, more close to the bottom slide of bottom side of the slide. And again, you can see the signal distribution for different type of targets. And again, we were trying to check different antibodies on the different channels. And also we have Akoya commercial antibodies as well as custom conjugated antibodies. And in the next few slides, I will show you the signal distribution for different targets. And you can see over here, it's the top tissue and bottom tissue and regions. Again, I was trying to extract similar regions from the top tissue and from the bottom tissue. Over here, you can see CD3E, it's Akoya commercial antibody, CD20, CD11C, this antibody was internally conjugated, pancytokeratin, CD31, CD45, CD68, HLA-A, and covalent one. So in the next slide, I will show you actually a combination of different targets, because obviously goal of multiplex assay is to able to detect different cell types. We didn't do any like deep analysis. That was not the goal of the study for us. It was important to implementation of the manual protocol on Barhelia spatial station. It's just a visual assessment. However, obviously you can appreciate the quality of the staining. Here you can see the structural components in bottom tissue in large and different regions. And in this slide, you can see region number 4. You can clearly see the vasculature and epithelial cells. And in the next slide, since we're working obviously with the tonsil, we would like to see different immune cells. And again, it's a bottom tissue enlarged different regions. And on the next slide, on this slide, you can see CD8, CD4, CD3, CD57 positive cells, PD1 positive cells. You can detect NK cells, cytotoxic cells, T helper cells. And here is just the overview of human FFPE tonsil staining quality. And we again perform the staining on Parhelia Spatial Station. You can see different cell types, epithelial cells, NK, T cells, monocyte macrophages, endothelial cells. Again, we were really satisfied with the staining quality. Also, I already mentioned that for a lot of us, H&E staining is also very important. And we wanted to make sure that after we perform Phenocycler Fusion, we can successfully remove flow cell and actually run H&E protocol on Parhelia Spatial Station. And we did this. And it actually was extremely easy to implement this protocol on Parhelia Spatial Station. Images was acquired on Fusion platform, and entire H&E protocol took for us only 20 minutes. And again, implementation of this protocol was extremely easy, and we can see that tissue integrity was preserved, and you can see that tissue staining quality on human tonsil. And again, I want to emphasize that it's a post-PCF H&E staining. And to summarize everything, I want to say that we actually were able to successfully implement Phenocycler staining protocol on Parhelia Spatial Station. We observed similar quality results when we compare visually manual staining and Parhelia Spatial Station staining. And we can see that we didn't observe any, Parhelia Spatial Station basically didn't compromise the quality of the staining, even when we used 2 tissue on the one slide, and the actual tissue was pretty large. It was covering a lot of microscopic light surface. And what is also we observe, it's a significant time saving, and we are using class reagents. And obviously, we're reducing the possibilities of the human error. And one thing that I think a lot of researchers can appreciate that Parhelia Spatial Station, it's an open platform. So you are not dependent on any reagents. You can do and you can optimize your protocols. You can implement the protocols of your need. You will use potentially less reagents. And I just to give to you example how I implemented Phenocycler protocol on Parhelia Spatial Station, but it's obviously a lot of different type of protocols that you can implement on Parhelia Spatial Station, and Naomi will give you more details in the next part of the webinar.
Thank you very much, Nadya, for not only going over the PCF protocol and how it was implemented on the spatial station, but for the gorgeous images. So next, I would switch a little bit gears and have a somewhat different perspective of the implementation of automation to different spatial workflows. And I will give you a few examples where we saw significant increase in efficiency or reproducibility, or when we could do something that was not possible before. We are lucky to have the Leica bond, the Omnistainer, and the Barhelia Spatial Station that I will refer to as PSS in our lab. And I will do a comparison of the major features. Then I will go to panel mixing for Phenocycler Fusion on the Spatial Station. Then I would like to give a few examples for the pre and post assay HNA staining for Visium, Phenocycler Fusion, CosMx, and Xenium technologies. Then I will try to give a few examples of automation for the multiplex IF assays that are run with amplification, including in-situ-plex, signal star, and Doppler staining. Then I would like to show you results about testing the image-ready cover pads for multi-cycle assays and touch on the DVEX and antigen retrieval. But who are we and what does our core do? So I am part of the Spatial Imaging Technology Resource that is part of the Office of Science and Technology Resources at NCI. We are located in Bethesda on the main campus. And our main goal is to evaluate, develop, and implement cutting-edge, mostly antibody-based proteomic analysis technologies, and recently spatial protein and RNA detection methods to facilitate discovery and translational research at CCR. Historically, we were offering 6 different methodologies, including automated capillary immunoassay or Luminex multiplex ELISA. But right now we are focusing on spatial technologies, and we have 3 of them available at the core. The first one is Phenocycler Fusion, as you heard, that enables detection of 40 to 100 proteins in a single tissue at single cell level. And we were beta testers for the technology in 2018. For a while, we had 2 systems operating in our laboratory. And right now, we have the old CODEX system as well as the Phenocycler Fusion. The other technology that we offer is GeoMx DSP that enables a regional detection of hundreds of proteins and thousands of RNA transcripts in tissues. We offer this service with collaboration of the genomics core, and we have both readouts, the Encounter as well as the NGS sequencer readout. What is special in the way how we offer this technology is that we customize morphology markers, and we also offer image analysis on the whole slide, not only the regions of interest that comes with the NanoString platform. Our newest technology is the CosMx SMI that enables single cell level detection of thousands or 6,000 RNA transcripts and about 60 proteins. This effort is led by Maria Olga Hernandez from our lab. And for this technology, we were also early users since 2022. And after all kinds of troubles for hardware and software, basically, they were all solved at the end of in November last year. And currently, we are running the 1,000-plex RNA panel routinely, and we are looking forward to implement the 6,000-plex panel and the protein panel. On this next slide, I would like to have a kind of a visual summary of the increasing number of spatial assays for detection of multiple proteins and transcripts. What you see here, it's basically the technologies that enable detection of proteins are in green. The ones for RNA are in yellow, and and some of them can do both. And here you see kind of divided in how many targets they can detect. It's again a visual, but on the next slide I will show you that many of these spatial assays have workflow automation already implemented. And I will talk a little bit about in-situ-plex signal star and opal assay, a little bit about the CODEX, although you had a very nice presentation from Nadya. And I will talk a little bit about the pre and post H&E staining of the other technologies. So as mentioned, we have the Leica-Bond, the OmniStainer, and the Parhelia spatial station in the lab. So let me just share with you how we think about and how we compare these technologies. For default protocols, all three of them come with protocols available, and often these are using the same kit, only that they apply multiple washes or double add of different reagents. Sometimes, for example, for RNAscope, the kit is different when it goes under automation versus manual. One question is always, what if you want to customize a protocol? For Leica-Bond, you can basically you can write the code. For OmniStainer, if you want to change, steps in the protocol, then you have to modify a very well-structured Python script. What you can do by yourself, or you can reach out to Parhelia to do it for you. When you set up the OmniStainer, basically you can select the number of the samples, different temperatures, different volumes that you want to add. But if you want to have a more major modification, again, you have to go to the script. For Parhelia spatial station, we have an interface that is shown on the top of the machine, that it's very easy to modify in a user-friendly interface. You can add steps, remove steps, skip steps, change pipetting speed, and so on. Again, from the course perspective, what about if there are errors during operation? So the Leica-Bond, when we see a few errors, then the protocol completes and we can see the errors in the log, but we cannot do much about it. For OmniStainer, the protocol stops, and again, we can see what was the step that went wrong. For the Parhelia spatial station, what is very nice, that the protocol stops, but it waits for the action from the user. So we can kind of rescue an experiment when we had an error. One nice feature is liquid sensing, and that one is available in the Leica-Bond. Is not available on the OmniStainer, but it's available on PSS. And the implication there is that you can choose to have a liquid sensitive, and it means that, if the liquid, whatever reagent is not enough, the robot stops and asks for action. But you can also disable if you kind of, you are sure that you have already the reagents and you don't care. Onboard reagent mixing, it's not available on Leica, but is available on both Parhelia stations. And you heard from Nadya that is very important when you have to prepare reagents fresh. The number of slides on Leica-Bond is 3 × 10. And it doesn't allow for staining of cover slips. On OmniStainer, for now, we can stain 12 slides at a time, and also we can stain cover slips, and that one is relevant to the old CODEX users. And the Parhelia spatial station right now enables for staining 12 slides, and currently we don't have the cover slip staining option. You heard from Jonathan that the temperature control is much wider for OmniStainer and Parhelia spatial station. Basically, we can go from 4 to 100 degrees, being able to control room temperature. Leica has temperature control only above 35 degrees. That volume, that influences reagent cost. It's significant, multiple hundreds of microliters on the Leica-Bond, but for OmniStainer or Parhelia spatial station, it's minimal. The operational cost, probably Leica, that everything is expensive. If something expires, you cannot use it. While on OmniStainer, you can use other than the tips, you can use almost any kind of consumables. And I want to mention that, if you want to use liquid sensing tips for perihelion station, that one is somewhat more expensive. Barcoding, it's required for the Leica band, and currently it's not available on the Omnistainer or perihelion spatial station. And all three of the instruments have service contract, for yearly maintenance. Okay, let's go to panel mixing. That it's really a desired feature for all workflows that involve pipetting of multiple reagents, especially in small volumes. And some use cases can be the Phenocycler Fusion for preparation of the reporter plate, as well as the antibody panel. Or we haven't done that, but we are thinking about that. Probably is the same for imaging or flow cytometry. Reporter plate mixing protocol has been, it is available on PSS and has been tested. I will show you examples from that. And the antibody mixing is currently being evaluated. What you see here is one of the modules, which is the P54 container, where you can put and store your reporter tubes. This one is on the left. And you can also see that, it is covered, so it prevents evaporation. And this module enables reporter preparation for 18 cycles. On the right, you can see that the caps of the tubes are kept separately. And, here I just indicated that these three reporters are added in one cycle, and we can arrange and have the robot programmed to have the panel mixing. On this slide, you can see what is required for this protocol. On the top, it's the control panel where you choose your protocol. At the bottom, you see the lever setup, and again, the P54 for the reporter tubes. Here, you see the black reporter plate. On the back, it's the container for the reporter buffer, and we use two kind of tips, the 50 and the 300 microliter tips for the pipetting. And there is a very easy to modify HitPic file to define the source, location, the destination location, as well as the volume. So we have run some testing on a tonsil sample. The tonsil sample was stained with a bunch of antibodies, and the visualization was done on the Phenocycler Fusion or with reporters that were prepared manually or that was prepared on the PSS. On the top, you see representative images for different signals that were detected after manual reporter preparation. In the middle, in yellow, you can see after preparation on the spatial station. And here we did some quantification. This one is intensity curves that we get when we are using Halo. The PSS prepared reporters are in teal, while the manual ones are in white, and you can see that the intensity curves, not only the visual representation of the images, are very much indistinguishable. So we can feel free to use the PSS for reporter plate mixing. So next, I will show a few examples for pre and post assay H&E staining. And for the different spatial assays, it is desired to have an H&E staining on the same slide. For tissue annotation, generally pathologists, they don't necessarily like to look on multiplex IF images. They want to look at H&E. So for Visium, being Visium or Visium HD, a pre-assay H&E staining is used to select different tissues, tissue regions. While post-assay H&E staining is performed for multiplex IF, CODEX, CosMx, or Xenium. The quality of the H&E staining varies depending on the tissue preparation, if we used or not antigen retrieval. And you can see here an example where you have DVEX and antigen retrieval on the left and DVEX only on the right. And it's hard to see, but there are less details and more damage to the tissue when we are using antigen retrieval. And it also depends on previous stainings. If you used multi-cycle staining, maybe cross-linking and imaging steps. Some of the samples stay in the machine for weeks. CODEX can stay on the machine for 2 days. CosMx can run for a week. I would like to make one point that these different protocols, they use different reagents. I don't know how not ignorant or knowledgeable are you about that, but hematoxylin can be a Myers or a Gilles hematoxylin. Eosin can be eosin Y, it can be an alcoholic eosin. And blue ink solution is usually the same. On this slide, I would like just to show that the different protocols use different reagents as well as different washing and staining steps. And on this slide, it's just for illustration that, for example, the Previsium H&E protocol is different. It doesn't use any ethanol washes. The post-xenium H&E protocol, it's longer. And for PCF or multiplex IF and CosMx, we are using the DACO H&E protocol. This one is just an example, to compare H&E with a signal star image. Or on the right, you can see it's an H&E image on a tissue that was run on PCF. And here on the next slide, there are a few examples of H&E staining for that is Previsium H&E, top lower magnification, bottom higher magnification. Post CosMx, the tissues are a little bit, more destroyed, and we have a post-xenium H&E. Okay, so right now I am moving into automation of multiplex IF assays that include the in situplex, the signal star, or the Opal IHC. And on this slide, I would like just to show that, they are all using amplification, but in different ways. And that one, of course, generates different problems in the, well, not problems, but different issues in the protocol. The in situplex, in the in situplex, we are using all barcoded antibodies that are added in a single step. There is a rolling circle amplification of all signals. And then in each cycle, 4 reporters and the nuclear staining is visualized on any scanner. For a signal star, all the antibodies are added in a single step to the tissue. They are also barcoded. Then we are amplifying 4 antibodies in each cycle. It's visualized, and then at the end of the round, basically the amplified signal is cut with double-stranded DNAs. This protocol includes close to 100 steps, and one challenge is that highly viscous solutions are used for amplification and antibody binding. The other assay, it's Opal IHC, when it's again a multi-cycle assay when primary, secondary antibody is used in each cycle with one of the Opal staining deposits. And the challenge here is that in each cycle, there is repeated antigen retrieval to get the next set of the stain there. Also, this protocol it has about 200 steps. But I would like to mention that all 3 protocols are available, implemented on the OmniStainer or the Spatial Station. Let me give you a few examples, a few images. This one is an 8-plex signal star assay that is implemented on the Spatial Station. Again, it has 2 rounds here. On round 1, I am just showing 2 images, CD3 in red and CD20 in green. For round 2, I am showing it's what you can see here is the DAPI staining, the CD8 in magenta, and the CD68 in green. For this, this was run on PSS with chamfered cover tiles. That was removed to have a regular cover slip, and it was imaged on a Zeiss Axio scanner. Another example is the UltraView assay, and in this case we were staining 3 tonsil samples on the same day. We used the PDL1 assay. We used again the regular cover pad that was removed to be replaced by regular cover slip, and it was done on Axio scanner. And not only that, you can nicely see the very similar staining on the UltraView assay, but when we did quantification of the signal at single cell level, you can see how comparable they are. Next, I would like to talk about the image-ready cover pads that by not having to remove them can prevent tissue damage when we apply multiple rounds of staining. Here on the left, you can see the regular cover pads that have a spacer facing the tissue. It has a 45-degree surface that helps with retaining larger volume. And these have to be removed and replaced with regular cover slips for imaging. While on the right-hand side, you can see the image-ready cover pads that are modified size 1.5 cover slips. They have the 70 micron spacers facing the tissue, and then another spacer facing outside to help with retaining the solutions and prevent overflow. And these ones are staying on the tissue during all the staining and imaging. Just an example for testing the image-ready cover slips, what you see here, it's a PDL1 assay. On the top, we used the chamfer cover slips that were removed and replaced. On the bottom, 2 sets of images, an image-ready cover pad was used with the Parhelia's mounting media. You can see that without quantification, the images are really very similar to each other. And I think you can see this. And again, one last topic is the DVEX and antigen retrieval. That can be really tricky. Usually we don't pay attention, you know, when we don't have difficult images, but we had worked with the TMA that was embedded in deep parafilm to protect RNA and protein quality. And when we were running conservatively the manual protocol for Phenocycler Fusion, basically we could not remove the paraffin completely. But when we were applying after the manual protocol, we were adding Parhelia's DVEX solution in the protocol. Then we got beautiful PCF staining on the slides. And this one is just to show you with 4 markers how beautiful the TMAs are. So what in the slides, what I have shown, I showed that the workflows for multiple spatial assays have been successfully implemented on Parhelia robots, being the OmniStainer or the spatial station, and are used to increase efficiency, throughput, reliability, and reproducibility of the work that is on the core. And this one is my thank you slide. And thank you very much for listening.
So I'm going to take back control now. Thank you very much to Nadya and to Noemi. Really great presentation here. We will be making this presentation available via PDF for all of our attendees. So if there is something you want to look back at, that we could potentially send that out to you. I did want to get to some questions, and I do have several, and I'll try to call out. Let's see here. Oh, this is a great one. Nadya, this one is for you. It says, based on your experience in working with Parhelia's technology, what was the biggest gain from automating the technology on the robots?
Well, as I already mentioned, it's obviously a few advantages. Most crucial one, it's a time saving for me that honestly at that time I can do QC of my data, or if you run your experiment like overnight, you can just go home, relax, do whatever you want. Plus, more consistent results. It's much easier actually to train people on automated platform rather than explaining entire like a manual protocol. And obviously reagent savings, that was quite crucial as well, because multiplex experiments are quite pricey.
Fantastic. All of those are expected. I really appreciate you answering that. Let's see here. I've got a couple of questions here. This one from Cesari. I think we've actually interfaced a little bit. Can this process be automated the same way for older version of the CODEX instrument, one that works with samples on cover slips? So I can actually give a little bit of insight there. Noemi's an expert at it. She actually has the capabilities in-house on our Omni stainer. So Noemi, I'll let you kind of take that for that. And I will say that this is something that's coming on the spatial station. It's a little less, we get a little less interest in that, but it is something that we are planning to offer and we can offer. Noemi?
Yes, and again, we have the Omni stainer that enables cover slip staining, and this is how we became interested in Parhelia's automation, because I was looking for something that can stain cover slips. So yes. And I think that Parhelia is really changing and kind of looking into implementing cover slip staining on the other platform as well.
Correct. Again, we've just now launched the spatial station in April of this year. And we're building it out as fast as we can with new assays, as we're seeing. Like I said, the new Signal Star kit from Cell Signaling Technologies is getting online. We've got a number of others that are coming online as well. So more from that platform very, very shortly. I have a question here from Thomas at Oregon Health and Science University. Can you use PBMCs for making slides and staining in place of tissue sections? So this kind of gets more to flow. And what I would say there is this something that we've looked at and we feel pretty confident we can do? And what I would tell you is in regards to that, stay tuned. We do have some things that we'll be launching in the future in that realm. Are they available today? No, they are not, but they will be. And Noemi, with your expertise in that realm as well, anything you can add there or anything you have to add?
I don't know if you wanted to stain PBMCs and run on infects, or you wanted to stain them, as slides, because that one is also doable. Not that we have done it, but theoretically, we were thinking of staining blood smears and PBMCs on tissue sections.
I wanna add to this. It can be done for Phenocycler Fusion platform if you wanna make a smear, and after that fix your cells, apply the flow cell. That's not a problem. Yeah. Oh, and obviously stain with your antibody before. Yeah, that's not the issue. Yeah.
Perfect. So let's see. We have a question from Tanya at Ohio State University. How practical is this assay for hospital clinical lab and not only just research use? What I can say there is that Parhelia did, in fact, design this robot to eventually move in that direction, and we have a lot of the auditing features and ISO features that you would need to do that. To do like a lab developed test or something along that lines. However, again, we stay agnostic to that. So if that's something you want to place that instrument in that environment and go through all the regulatory checks, then those things are there, including there's reporting at the end. and different audit features that you can implement as well. Let's see here. There's quite a few questions actually, which is great. And I'll let Nadya and Noemi answer this one. Does Spatial Station or OmniStainer allow you to optimize protocols easily? What variables can I control to optimize assays for tissue types?
If I just, again, use this slide, protocol customization used for optimization. Of course, you can add steps. You can increase pipetting speed. You can change the time for different steps. This is what I think, or how I think about protocol optimization based on your needs.
And yeah, if we are talking about, for example, Phenocycler protocol, as you can see, people are using different timing of antibody incubation at a different temperature. You can obviously do overnight staining at 4 degrees with a thermoblock on, or you can do like 3-4 hours at room temperature, or if you want to expedite your study, you might try to do it 1 hour, 37. It's really up to you. Plus, it's very easy to switch, at least in Parhelia Spatial Station, between fresh frozen and FFP protocol. You just disable and enable a couple of steps, and that's it.
Fantastic. And apologies, I said Ohio State for one of the previous questions. It was Oregon Health and Sciences University. Apologies. Football season, that's where my head was. Let's see here. This is a really common question, actually. So the question is, how long are the protocols? Is it faster than doing it manually? Very, very common question. I'll let the experts answer, but what I would say is that you save time in the sense that you get to walk away from the robot. But we follow the protocols to a T based on the manufacturers of the chemistries and the imaging. So we try to follow it as close as we can. You do save a little bit of time probably with the pipetting of the robot being a little faster, especially if you're doing a panel mix where you're doing something high plex. But I would imagine we're following along the protocols. Nadya, Noemi, is that your experience?
I would not say that, you necessarily save time. And protocols really depend, number of steps in a protocol really depend on the assay itself. Again, InSituPlex has just a few steps. SignalStar has around 100 steps, while Opal around 200. In my experience, it's not necessarily that it's faster compared to manual, but when you set it up, you don't have to babysit it. You are free and you come and pick up the slides what are ready to go for imaging or for whatever next steps you want. This is how I see, the advantage of having the robotic system.
And I want to say, for me, when we were basically transferring manual protocol to Parhelia Spatial Station, and comparing the manual staining to the staining from Parhelia Spatial Station, it was quite important that we are, in a way, following manual protocols. And all these manual steps, that they are exactly, in a way, the same, but it's just that a robot is doing it. Again, I would say from the time savings, it will be for PCF, for example, exactly the same. But again, you don't need to, as Noemi said, babysit the instrument.
Fantastic. Thank you for that. Let's see here. Looks like we have one more. If we have any other questions that people want to have us answer, have them go ahead and put the questions in. I do have one more. This one is for Nadya from University of Minnesota. It says, what was the reagent savings, or how much reagent did you save on running a PCF study on Spatial Station versus manual runs?
Okay, so as I think I mentioned before, on each step, Parhelia Spatial Station required 150 microliters, but you want to keep in mind also that when you are aliquoting your reagents, you will be aliquoting a little bit higher amount, but obviously it's much, much less comparing to the regular manual staining protocol.
Fantastic. Fantastic. Thank you for that. And with that said, we're gonna go ahead and wrap up. So again, thank you to our speakers, and Nadya and Noemi. Fantastic presentations. The images are quite beautiful. I love looking at them. They're just like artwork. They're absolutely gorgeous. So really appreciate your insights and your ability to share your insights with the community here. Again, thanks to LabRoots for hosting us and allowing us to have this webinar with you. And then thanks to all of our attendees. Really appreciate you joining. Please stay tuned. We're going to do a lot more of these with Parhelia, different applications, different sort of protocols, and new features of what we're bringing to the market. Again, we're hoping to revolutionize the sample prep for all of your spatial biology needs with push button precision. Thank you so much. Have a great day.
Thank you, everyone. Bye.